Welcome everyone, I'm Minh Bao Huynh, and I'm going to talk about some specialized polysaccharides which you can find in tissue homeostasis and diseases. Before I'm going to do this, I would like to thank the organization committee for the invitation for this Latin American Glycobiology Congress. I am a researcher at the Gly-CRRET laboratory, which is in France and which is at the University Paris-Créteil, under the direction of Professor Dulce Papigacia. Here we have specialized facilities for our glycemic research, like the glycomics platform, whereas we do the structural and functional studies. We have also associated facilities like cell culture, image microscopy, animal housing, histology, and radioactivity facilities, whereas we do our glycemic research on diseases. And all together, with the help of these facilities, I'm going to present you the case of Alzheimer's disease. So Alzheimer's disease is a slow or fatal disease of the brain and a major cause of dementia with aging as the major risk factor. This disease has been discovered by Albert Alzheimer when he observed his first patients back in the 1900s. Alzheimer's disease has been characterized by two proteins. We have the A-beta and the tau protein that accumulates and aggregates in the brain, forming these extracellular amyloid deposits and intracellular neurofibrillary tangles. When we look at the sequential change in measures of Alzheimer's disease, we see here in the red line the A-beta and the amyloidogenesis. Over time, this disease progresses, and we see here in the blue line the tau protein and the tauopathy. For this presentation, I would like to focus more on the tauopathy. We have the tau protein, and normally this tau protein becomes phosphorylated and stabilizes microtubules. In pathological condition, tau protein becomes hyperphosphorylated, abnormally phosphorylated. It will accumulate and form tangles, destabilizing the microtubules. Over time, this tauopathy is killing and compromising brain cells and causes, for instance, the loss of memory and cognitive decline, with worldwide millions of patients that are affected, and every 3 minutes there is one new case. Alzheimer's disease is also the fourth leading cause of death and the first cause of dementia. Besides the known hallmark, I'm going to present you a new hallmark. We call this the glycemic hallmark, and that contains intracellular Heparan sulfates, which are known already in the 90s to co-localize with tangles. I show you another example from an upcoming publication that we are going to publish in Reberg et al., whereas we see also co-localization of Heparan sulfate with tangles. And these Heparan sulfate play a role in events even before the classical hallmarks, targeting neurons, making it that we have neuronal Heparan sulfates playing a critical role in AD-related tauopathy. So besides that Heparan sulfate can co-localize with tangles, it is also known that the tau protein hyperphosphorylation and aggregation is dependent on Heparan sulfate. Heparan sulfate can induce conformational changes to the tau protein to expose new phosphorylation sites, and these are all examples to show the importance of Heparan sulfate and the presence of Heparan sulfate in tauopathy-related AD. In general, Heparan sulfate have essential roles in biology. The question now is, what are these Heparan sulfates? So Heparan sulfates belong to the family of glycosaminoglycan, and a glycosaminoglycan is a long chain of linear polysaccharide which can be attached to a core protein forming a proteoglycan. They are composed of disaccharides, which can be formed of an uronic acid that contain a glucuronic acid or an iduronic acid, and an hexosamine that can be an N-acetyl glucosamine or an N-acetyl galactosamine. The most important is that they can be sulfated at the different positions indicated here in red. Based on their disaccharide units, we can distinguish different GAG families. We have the heparan sulfate and heparin, we have the contin sulfate A to E, and keratan sulfate, which are known to become sulfated and also are known to be attached to a core protein. In fact, there's an exception, and that is uronic acid, which is not sulfated and which is also not known to be attached to any core protein. So these glycosaminoglycans and also their proteoglycan form can be found located at the cell surface and also in the extracellular matrix. When we look to heparan sulfate and heparin, these type of glycosaminoglycans are very highly sulfated. Heparin has the same building blocks as heparan sulfate, but is even more sulfated. Based on their sulfate pattern, heparan sulfates are very complex polysaccharides. The heparan sulfate structural complexity can be found back during its biosynthesis by a battery of modification enzymes. And when we look to the heparan sulfate biosynthesis, we can find a core protein with a serine residue. To this serine residue, there will be O-linked acetylose, galactose, and glucuronic acid. This is also known as a tetra linkage. And at the moment when there is an N-acetyl glucosamine attached, the heparan sulfate chain becomes formed, followed by a glucuronic acid, N-acetyl glucosamine, glucuronic acid, N-acetyl glucosamine, and so on. So in the same way, the disaccharidic units are formed, and during the elongation, many enzymes are implicated. In the same time, the heparan sulfate chain can become modified. In fact, what happens is that the heparan sulfate chain becomes sulfated. This occurs by different enzymes. We can have the NDSTs, which can give N-deacylation and N-sulfation. We can have the epimerase. Epimerization occurs with a conversion of glucuronic acid into iduronic acid. And then we can have in the chain at different positions sulfation at 2, 6, and 3O position by different sulfotransferases. The heparan sulfate chain can be divided in different domains. We can have the Na domain and acetylated domains, which are domains which are not sulfated. We can have domains which are partly sulfated. We call them the Na and S domain. And we can have domains which are highly sulfated. We call them the NS domains. So when we look to heparan sulfate and all these domains based on their backbone and disaccharides, heparan sulfate can raise specifically sulfation patterns which differs from one heparan sulfate to another. And these specific sulfation patterns can be associated with many different factors that can bind specifically to heparan sulfate. Here you have an example of some sulfation patterns binding to particular ligands, for instance, like the VEGF and the FGF, vascular endothelial growth factor, or basic fibroblast growth factor, which can be also located in the extracellular matrix and on cell surfaces, making that these polysaccharides are key regulators of protein function. So it makes it that these proteoglycans and glycosaminoglycans, which can be located at the cell surface or in the extracellular matrix, play essential roles in the cell function. And they are involved in biological processes, for instance, like neurogenesis, neurodegeneration, tumor growth, wound healing, development, and many others. However, few is known about these gag structures and function importance in most of these processes. When we look to heparan sulfate structures, and especially to the NS domains, a domain where we have highly sulfated heparan sulfates, we can find 3O sulfation. So this 3O sulfation is a very rare type of modification within the heparan sulfate chain, modified by 7 different isozymes. In overall, when we look to this heparan sulfate, we ask ourselves the question whether we can find a fingerprint for a particular disease. Within this, we ask the question, can we have alteration of this heparan sulfate, for instance, in the sulfation level? So that means when there's pathology, can we have more or less sulfation? And can we have alteration of heparan sulfate based on their sulfation pattern? So we also call this sulfation code. So to make it more difficult for you, so is it possible that during physiological condition going to the pathological condition, is it possible that we can have both the situation? So central in our study, we asked the question, how would Heparan sulfate alteration affect the biological function and diseases? Over time, and with the help of genomics and proteomics, but also glycomics, or the combination of all three, we gain novel insights in the heparan sulfate biosynthetic modification enzymes. So when we look to the biosynthetic machinery of heparan sulfate, we can see that there are 51 main genes, which we can find here in this table, also published by Owidia et al., and you can find this back in medRxiv. So we can have genes which are wild expressed, and we can have genes which are tissue restricted in expression. We can also have genes which we can cluster as essential genes, and we can have genes which are non-essential. In fact, there are two main types of heparan sulfate. We can have ubiquitous heparan sulfate, which are widely expressed, and we can have specialized heparan sulfates, which are tissue restricted and carrying specific sulfation codes, including the 3O sulfation. So within this, we asked the question whether 3O sulfation or also 3-SHS can be involved in the heparan sulfate accumulation in cells during the tauopathy. In Alzheimer's disease, we saw that heparan sulfate changes in the structure and composition. Here we have a result that we published in 2019, whereas we took human postmortem delay hippocampal tissue and we extracted glycosaminoglycans and also we quantify them. Here we have a result that shows that in Alzheimer's disease, we have an increase of total sulfated glycosaminoglycans. And this increase can be correlated to an increase of heparan sulfate. We also saw that in Alzheimer's disease, the heparan sulfate had strong binding capacity with the tau protein. And overall, we have in Alzheimer's disease a change in structure and composition that could be related to a change in the quantity and the quality of heparan sulfate. So it was very interesting to also to study the modification enzymes from heparan sulfate, and especially the enzymes that could be involved in the making of the 3O sulfation. So in human, there are 7 different isozymes, and these are the heparan sulfate 3O sulfotransferase 1, 2, 3A, 3B, 4, 5, and 6. It was known that heparan sulfate 3O sulfotransferase 2 and also 4 were predominantly expressed in the rat and human brain. In 2011, we published that human heparan sulfate 3O sulfotransferase 4 shows significantly increase in expression with age, but not heparan sulfate 3O sulfotransferase 2. In fact, we were dealing with specialized and tissue-restricted sulfotransferases. And here we asked the question, can these specialized and tissue-restricted sulfotransferases that makes these neuronal 3O sulfation, so 3-SHS, can they be a target for Alzheimer therapy? When we analyze the expression of the modification enzymes from the heparan sulfate biosynthesis in Alzheimer postmortem tissue, we saw that heparan sulfate 3O sulfotransferase number 2 was very highly expressed, making us to target this enzyme. Here I show you an example of a zebrafish model that we published in 2015. These fishes express the P301L mutation in the tau protein, representing frontal temporal dementia. And these transgenic fish express AD's red. So you can see here the red fluorescent. And at the moment when we stain these fishes with an antibody recognizing tau, you can see also green fluorescence. So when we target the enzyme heparan sulfate 3-O-sulfotransferase 2 in these fishes, we saw that the tau protein disappeared in these morphins. Additionally, we also published that the 3-O-sulfate heparan sulfate can accumulate at the intracellular level and interacts with the tau protein to induce tau abnormally phosphorylation in cells. We saw that heparan sulfate 3-O-sulfotransferase 2 and its product, the neuronal heparan sulfate, the 3-SHS, play a critical role in the AD-related tauopathy. So during our glycolytic journey, we saw that in Alzheimer's disease we had an alteration in structure and function of heparan sulfate. In fact, heparan sulfate changes in quantity and in quality with implication of 3-SHS. And these 3-SHS accumulates at the intracellular level with interactions with the tau protein and induces tau abnormal phosphorylation. We also saw that heparan sulfate 3-O-sulfotransferase 2 play critical roles for AD-related tauopathy. So we were dealing with very specialized heparan sulfate structures and was needed for further investigation. So our following up studies were to investigate whether the 3-SHS by the heparan sulfate 3-O-sulfotransferase 2 can promote the spontaneous self-aggregation of tau in cells. Furthermore, to investigate in human brain the expression of the 3-SHS and the enzyme. And we were aiming to investigate the mechanistic action of the 3-SHS and the enzyme. So for the moment, I would like to go a little bit in more detail concerning the first part, whereas we have recently a publication. Here we were able to construct and stable transfect HEX cells with the enzyme and tau or mutated tau or the combination of the enzyme and both the taus. This work could be realized with the help of Dr. Denis Bier from the CIEA, and also there's Dr. Nicolas Reberg, who recently received his PhD. So in this particular order, I'm also going to present you the results. So we have here wild-type cells, HEX cells, then followed by transfection with the enzyme, or with the tau, or the combination enzyme and tau, and then we have cell lines that were transfected with the mutated tau, and cell lines were transfected with the mutated tau and the enzyme as control. We looked to the HEC wild-type cells which were not expressing the tau protein or the enzyme heparan sulfate 3-O-sulfotransferase 2, but they were expressing heparan sulfate 3-O-sulfotransferase 1, 3A, 3B, 4, and 5. So this was with the transcriptional level based on qPCR, and these cells were perfectly for the expression of tau or the enzyme heparan sulfate 3-O-sulfotransferase 2. And when we look to the protein level based on Western blots, we saw that the enzyme was expressed. When we looked to the immunocytochemistry, we saw also that the enzymes were expressed in green in the cell lines that were transfected with the heparan sulfate 3-O-sulfotransferase 2. And not only that, they were also expressing the product, which you can see in red. Here you can see also confocal images of the product, the 3-SHS, which indicates that we have an overexpressed cell line with an active enzyme. So we not only overexpress the enzyme, but also we overexpress cells with the tau or mutated tau or the combination of both the enzyme and the different taus. Here you see a result of the qPCR on a transcriptional level, the expression. Also you see a gel electrophoresis. And what do we see? We see that every cell line that have the enzyme, it expresses the enzyme, and every cell line that have tau or mutated tau, it expresses the tau. In the same way, we were looking at the protein level based on Western blot to see the expression of the enzyme, and every cell line that has the enzyme express these enzymes, and also to see whether these cells could express the tau, and every cell line that have the tau or mutated tau expressed the tau. So when we look to the immunocytochemistry, in fact, we saw the same. We saw localization of heparan sulfate 3-also-transferase 2 with tau, and in general, these results show that the enzyme and tau do not induce their reciprocal expression. Now, the question is, can these cells undergo a tauopathy and making aggregates? Here you have a very nice illustration that I took and I modified from a NUN, showing that we can have the tau protein hyperphosphorylation and abnormally phosphorylation causing accumulation of this tau and also destabilizing the microtubules, followed by where you have monomeric tau, oligomeric tau, perhelican filaments, and neurofibrillary tangles. For our publication in Scientific Report, we were looking to whether these transfected cells with tau and heparan sulfate 3-also-transferase 2 could make oligomers, and we could detect that with a particular antibody. Here you have an immunohistochemistry result again, whereas we use an antibody that could recognize total tau, and you see all the cell lines that could express tau or mutated tau were expressing this total tau. And then at the moment when we were using an antibody T22 that could detect oligomeric tau, we saw only in the cell lines that were expressing the tau and the enzyme heparan sulfate 3-also-transferase 2, or mutated tau alone, or mutated tau with enzyme, that there was oligomerization. Here you have the quantification of the ICC and also a Western blot with its quantification, showing that we have oligomeric tau that was significantly increased in these HEX cells expressing the enzyme and tau, or mutated tau. So overall, these results show that heparan sulfate 3-also-transferase 2 could trigger the cell autonomous oligomerization of tau and mutated tau. So we were also looking to phosphorylation of tau protein, and we took cells, we were extracting these cells in RIPA, and we did Western blots. Here you have the Western blot and also its quantification, and what we see in this Western blot when we use the anti-tau antibody that recognizes total tau, we see that the cell lines that were expressing the tau express the tau with a size of 64 to 68 kilodalton. So when we use saccharosylate extraction with high concentration of salt, which allow to recover oligomeric tau, we saw with different antibodies detected to recognize total tau, oligomeric tau with T22, or with PHF1 to recognize phosphorylated tau, that these cells express under slight denaturated conditions or complete denaturated conditions tau at 64 to 68 kilodalton, and this was only for the cell line that was transfected with the heparan sulfate 3-also-transferase 2 and tau, mutated tau, or the enzyme with mutated tau. 64 and 68 kilodalton in tau are hyperphosphorylated tau. So we have heparan sulfate 3-also-transferase 2 that could promote tau and mutated tau oligomerization, but also hyperphosphorylation in these cells. So we were also investigating whether the product of this enzyme, the 3-SHS, could have intercellular accumulation in these cells. When we look to figure A, we see HEX cells overexpressing the tau, and we took an antibody that could recognize 3-SHS and we labeled them, and you see that in red. So we also took an antibody that could recognize total tau, here in green, and you see tau localization. When we take an antibody that could recognize oligomeric tau, the T22, we did not see any signal. Figure B are HEX cells with the enzyme and tau overexpressed, and what we see here is co-localization of 3-SHS and tau, but also with oligomeric tau. Figure C is the quantification, and below we see confocal imaging with figure D, the HEX cells overexpressing the tau, and figure E, HEX cells overexpressing the tau and the enzyme. And what we see here is that we have internalization of the 3-SHS, which co-localize with tau and oligomeric tau. We see the same for mutated tau and mutated tau with the enzyme. And overall, these results show that we have 3-SHS that are internalizing and accumulating in the cells, produce the tau and the mutated tau oligomers. So we publish all of these in a paper here with all the authors, showing that the heparan sulfate 3-O-sulfotransferase 2 expression induces the cell autonomous aggregation of tau. I would like to go to the next part where we can have some previews from upcoming publications concerning the expression of the product and the enzyme in the human brain, and also its mechanistic action. One of the highlights concerning an upcoming publication by Rebecca et al. It concerns the special transcriptomics from the Alzheimer brain for human, but here I'm going to show you the mice brain for the different sulfotransferases. We have here the sulfotransferase 3-O sulfotransferase 1, 2, 3A, 3B, 4, 5, and 6. We see very specific expression for the enzyme heparan sulfate 3-O-sulfotransferase 2 and 4, whereas in B and C you see the different regions and also the different layers. And in D, we see that the heparan sulfate 3-O-sulfotransferase 2 express in the CA1 and the CA2 region of the hippocampus. So these results show that the heparan sulfate 3-O-sulfotransferase 2 can render specifically brain regions vulnerable to AD-related tauopathy. So this data and many others was possible with the help of Dr. Olivier Pape, who is also co-founder of the company Gly-CRRET. Here I show you another highlight from the paper Rebecca et al. That is under publication. So here we have again the aggregation and implication of the product and the enzyme based on Western blot. This you already know that we have soluble tau expression in the tau cell line, but also in the cell line with the enzyme and the tau expressed. And then we can have oligomeric tau expression. It's only in the cell line with the tau and the heparan sulfate 3-O-sulfotransferase 2, whether this is in the denaturated or slightly denaturated condition. And then what we also did is looking to the electron microscope to see the fibers. We also show that the 3-SHS induces aggregation kinetics, and we also show the single cell analysis, whereas we show that this expression of the heparan sulfate 3-O-sulfotransferase 2 express specifically in neurons, making that this enzyme can render specifically brain cell vulnerable to AD-related tauopathy. Then there's another publication upcoming from Gomez and Now, whereas we look to the mechanistic part and where we look to the 3-SHS internalization. So here you have some highlights of the study, and here you see a cell line is wild type HEC, whereas we stain and label them with the 3-SHS antibody, and we see very slight membrane staining, and then we have a cell line that has the tau P301S mutation representing frontal temporal dementia, and we stain them with the 3-SHS antibody, and we see that they internalize here in the conf call imaging. And when we take an antibody that only can recognize heparan sulfate, no 3-O-sulfation, we see that they stain very strongly at the cell membrane. In this study, it was very interesting to see the anisotosing pathway, and here you have the highlight, for instance, with the mutated cell line overexpression, whereas it shows that we have a co-localization of the calvulin pathway with the 3-SHS, and this was not the case for the clathrin pathway. Of course, this data is just an illustration, but we have them with the enzyme heparan sulfate 3-O-sulfotransferase 2. I'm going to end this presentation with a take-home message. We have heparan sulfate that are complex molecules and that have a battery or biological functions. We have neuronal heparan sulfate that play important roles in the brain with specific structures, the 3-SHS, made by the enzyme heparan sulfate 3-O-sulfotransferase 2, which can render specifically brain cells and regions vulnerable to Alzheimer-related tau pathology. So all these studies and insights will give new possibilities for Alzheimer's disease. In fact, the art of glycoscience translated into a cure. And here I take the opportunity also to present a company, Gly-CRRET, whereas we are able to stop Alzheimer's disease. Many thanks goes to the collaborators, for instance, in France, Dr. Bière, Dr. Cabani, Dr. Pousset, and Dr. Denat from the CEA. In the Netherlands, we have Dr. van Kupperveld and Dr. Damen to thank. And also we have a very strong collaboration with UNAM in Mexico and to thank Professor Centeno and Dr. Ramizé Hernandez. Besides the many collaborations that we have in many countries, we also would like to thank the companies, the Gly-CRRET and OTR3, and our financial supports. Many thanks goes to the facilities that we have in-house for doing the research. We have the cell culture coordinated by Ms. Josephine and Dr. Ivan. We have the image facility coordinated by Mr. Carpenter. We have the animal housing coordinated by Dr. Owija, and we have the histology coordinated by Dr. Lavier. We have radioactivity facility coordinated by Professor Papigasia, who is also involved in the glycomix platform coordinated by me myself, and I work together with Dr. Gomez Henao, who recently also got his PhD. Many thanks goes to the people from the lab with our 3 groups which do glycanic research on different fields, neuroscience, matrix therapy, development, and so on. Also, we are not only doing research, but we also do teaching. So many thanks goes also to the students, undergraduate students, PhD students. If you would like to have any contact, you can contact Professor Papigasia by her email or our secretary, Ms. Larusso, by her email. And then you have me. Many thanks for this congress. And if you would like to have any contact, you can contact me by email. Thank you very much.